All right, everybody, welcome back. Ready for another deep dive?
Always ready to dive in.
Well, today we're looking at some pretty cool research.
Well, yeah.
Yeah, it's all about antibodies
Nice.
and how hard it can be to find ones that actually work the way we want them to.
That's a huge field.
It really is, and it's important for so many different areas of medicine.
Absolutely.
So the paper we're looking at today focuses on these things called M&S antigens.
Okay.
They're basically like little markers on the surface of red blood cells.
Right. So they're important for blood transfusions and stuff.
Exactly. If you get the wrong blood type, your body can attack those antigens and cause a whole lot of problems.
Definitely.
So finding antibodies that can bind to these antigens could be a game changer.
For sure. It could lead to better ways to match blood types.
It could even help us diagnose certain diseases or deliver targeted therapies.
Wow. So the possibilities are huge.
They really are. So in this study, the researchers used this technique called phage display.
Phage display.
Yeah, it's kind of like fishing for antibodies.
I'm listening.
You have this huge library of potential antibodies, and you use a specific bait to try and find the ones that stick.
Interesting. So what do they use as bait?
They use different parts of these M&S antigens,
Okay.
and they were able to find antibodies that bound to some of those parts.
That sounds promising.
It does. Right, but here's the catch. None of the antibodies they found could bind to the complete M&S antigen on an actual red blood cell.
Oh, wow. So close, but no cigar.
Exactly, and that's the real mystery here.
Why would that be? If they bind to the parts, why not the whole thing?
Well, it turns out that these M&S proteins are more complicated than we thought.
How so?
For one thing, they often exist as dimers.
Dimers, like they pair up
Exactly, they're like two legs of a chair.
for stability.
Yeah, they need each other to stand up straight.
And that messes things up for the antibodies.
It can, because the antibody might need to bind to both parts of the dimer at the same time.
I see. So it's not just finding the right key, it's finding one that can fit into two locks at once.
Pretty much. And to make things even trickier.
There's more.
Oh yeah, these M&S proteins are also glycosylated,
Glycosylated meaning.
meaning they have sugar molecules attached to them.
Oh, wow. That sounds complicated.
It is, those sugar molecules can block certain binding sites or even change the overall shape of the protein.
So it's like trying to find a keyhole that's covered in vines and maybe even moved somewhere else entirely.
That's a great analogy.
Yeah.
So you can see why finding antibodies for these things is such a challenge.
Definitely sounds like you need a really detailed map.
And that's where computer modeling comes in.
So they're building 3D models of these proteins.
Exactly. And it's giving them some really interesting insights.
Like what?
Well, for example, they found that the single or monomeric form of these proteins,
Without their dimer partner.
right, it's much less stable than the dimeric form.
So they really do need that buddy system.
It seems that way. They also found that some M&S proteins have this unique structural feature called a Mott hairpin.
A Bach hairpin. What's that?
It's kind of a loop in the protein structure.
And not all of them have it.
Right. Only certain ones, which could explain why some antibodies bind and others don't.
Fascinating.
Yeah, and it really highlights just how complex these antigens are.
For sure, and how much we still don't know about them.
And one of the biggest challenges the paper talks about is replicating the natural environment.
What do you mean?
Well, it's one thing to study these antigens in isolation.
Like in a test tube.
Yeah. But it's totally different when they're on the surface of a red blood cell surrounded by all these other molecules.
Right, the context matters.
It does. And it seems like that context might be crucial for antibody binding.
I see. So we can't just look at the antigen itself. We have to look at how it interacts with its surroundings.
Exactly. And that's something that's really hard to replicate in the lab.
So where do we go from here if it's so hard to find these antibodies?
Well, the researchers suggest a few things.
Okay, I'm all ears.
One is to incorporate glycosylation into those computer models.
So add the sugar molecules.
Yeah, because right now, most models don't include them.
So we're getting an incomplete picture.
Exactly. By adding those sugars, we might be able to see how they affect antibody binding.
Like seeing the vines covering the keyhole.
Exactly. And they're also looking at new methods for developing antibodies that are better suited for complex proteins.
So proteins that are maybe a bit messier or don't have a clear shape.
Right, they call disordered proteins.
Interesting. So they're trying to find new tools for a new kind of target.
Yeah, because the old tools might not be up to the task.
It's like trying to catch a fish with a net that's too big.
Exactly. You need something more precise.
So this research is really pushing the boundaries of antibody development.
It is. And it shows us that even when we don't find what we're looking for.
We still learn a ton along the way.
Exactly. And those lessons can lead to even bigger breakthroughs down the road.
It's all part of the process.
It is. And you know what's really cool about this whole thing?
What's that?
The fact that these M&S antigens are so complex.
Which makes them such a pain to work with.
Right, but it also makes them incredibly interesting for developing really specific therapies.
How so?
Well, because if you can find an antibody that binds to just the right spot on that complex antigen.
You can target a very specific process or cell type.
Exactly, which opens up all sorts of possibilities for personalized medicine.
Wow. So instead of a one-size-fits-all approach.
We could have treatments that are tailored to each individual's unique biology.
That's amazing.
It is, and it's all thanks to the incredible complexity of these tiny little proteins.
So what do you think, listeners? Are we on the verge of a whole new era of antibody therapies?
Let us know your thoughts, and till next time, happy deep diving.
See you next time.